this. So this is occurring in a variety of fungi, including yeast. And we talked a little bit before, but the principle of making alcohol includes taking some water and some barley, putting a bunch of extra sugar in it. And then if you're making wine, you will close the container. And of course, if you add something called an air trap, and that air trap is going to prevent further oxygen from getting in. But when you first close it, there is some oxygen in there, and the yeast begin to respire aerobically, and they produce carbon dioxide, right, as the output of that cellular respiration. But eventually, they use up all the oxygen in the system. That the bubbles that you see at the beginning of making wine or beer are from that carbon dioxide being produced. But after a while, all the oxygen is used up, and now the yeast can only respire anaerobically. So the bubbles stop, and what happens is that instead of shuttling this pyruvate into citric acid cycle, and instead of shuttling the NADH into oxidative phosphorylation, that pyruvate is converted to acetaldehyde and then ultimately to alcohol in a process that allows you to recover your electron carrier so that it can perform more glycolysis. And you build up alcohol, and you can measure the amount of alcohol that you have in that container, and you stop when you have the right proportion for beer or wine or whatever you're making. Okay? That's alcohol fermentation. What's happening in lactic acid fermentation is also very interesting. Is I'm sure many of you know, we build up lactic acid in our muscle cells when we exercise really strenuously. And what's happening in this situation is that when we exercise so strenuously that we deplete the oxygen in our mitochondria, remember your muscle cells have lots of mitochondria in them, and they're prepared to do lots of aerobic respiration. But if you exercise really intensively, you use up that oxygen. Or it's not that you use it up, it's that you cannot breathe fast enough to replenish it fast enough. And your body still wants extra ATP. So it gets that by taking the pyruvate and turning that into lactic acid. And that lactic acid in order to recover the NAD+. And that lactic acid builds up in your muscle cells. And that feeling of stiffness that you have the day after is usually because of that accumulation of lactic acid in your muscle. Once you relax for a while, though, that lactic acid goes actually to the liver. And in the liver, your lactic acid is reconverted to pyruvate. And assuming that, and if you're, then that pyruvate can actually be used to enter the citric acid cycle now. So in our own bodies, this is not it's not a terminal state here. This lactic acid will be reconverted to pyruvate and can enter citric acid cycle. But it's just a mechanism for the body to generate extra ATP through anaerobic respiration. So those are two forms, they're called fermentation. So fermentation is simply a mechanism to allow glycolysis to continue in the cell in the absence of oxygen. And glycolysis, the goal of glycolysis is simply the recovery of your electron carrier. The goal is not this end product, right? These are waste products as far as they're concerned. Although in lactic acid fermentation, the body's able to utilize that lactate to convert it to pyruvate. Okay, so that's fermentation. There's another kind of anaerobic respiration, which I mentioned briefly, and I bring this up again primarily so you keep straight the terms. There's another kind of anaerobic respiration which occurs in some bacteria, and in these bacteria, they are going through glycolysis, and they are going through the electron transport chain, but the final acceptor of electrons is not oxygen. So it's called anaerobic respiration because it occurs in the absence of oxygen, but it's a little bit tricky to remember because we usually think of anaerobic respiration as fermentation. It's not always. Anaerobic respiration occurs to any process that occurs, any form of respiration that occurs in the absence of oxygen. Fermentation specifically refers to the maintenance of glycolysis and not going through electron transport chain. But there are bacteria that have anaerobic respiration that go through glycolysis and then go through electron transport chain. The only difference is here they are not using oxygen as a final electron scepter. They're usually using a sulfate or a nitrate. And there are a variety of bacteria that go through this that live under in anaerobic environments. So don't get those two things confused, anaerobic respiration and fermentation. Oh, I have a video of this. Can you hear something?
Volume is off.
What was that?
Volume is off.
Oh yeah. Thank you. It's going to go screaming now. We're going to turn this down.
Most ATP is made in the
So I
cell's
want to show you this one too.
mitochondria.
Wait a second.
ATP, how...
Okay. So I'm going to show you the fermentation one, and then I'll show you this one.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are two types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
Yeah, fermentation. So, I'm going to show you now an animation of cellular respiration. It's a little bit of a movie.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy and fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, one carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Yeah. Now, I know we only have a few minutes.